So, we will start now with our webinar, Prilling by Vibration and Spray Drying in the Biotechnology and Food Industry. I'd like to welcome you. My name is Willy Hüsler. I'm from Büchi. Since a few years, I'm in the international sales and responsible for some countries in Europe and some countries in Africa. Büchi has been founded in 1939. We have about 700 employees, work within 5 segments. Our turnover is about 97% on export, and we have over 100,000 customers worldwide. Our main segments are in chemical, environment, feed, food and beverages, as well as pharma and biotechnology. We work with the five segments. In So, sorry for that. In our pharma segment and chemical segment, we see, for example, our rotary evaporator, which is one of our main and most important products. Then preparative chromatography or flash chromatography, and as well the melting point apparatus. For developing new formulas, we are focusing today. We see an encapsulator, a spray drying, and a freeze dryer. Then food quality, we have there an NIR spectroscopy, then an extraction unit for fat determination, and also a Kjeldahl unit where you could, which could be used for protein determination. These all in quality control. Then process control, we have here a sample of our NIR online. So this is a process sensor. And then we have environment tests where we use parallel evaporation or parallel concentrator. They are accelerating the sample throughput. And last but not least, we'd like to have our slogan, in your ears, quality in your hands. Now, something about the rules. We would like to have the questions. If you have questions at the end of the entire session, please write them into the questions area. And at the end, we will answer them either by word or by writing, depends a bit on the complexness of the question. So now I'd like to introduce our distributor from Ireland, Mr. Michael Kerwick. Michael, would you unmute yourself and start the introduction from your side, please? Okay. We have a technical issue with the audio setup from Mr. Kovic. I would therefore introduce Michael Whelan, which is our first presenter, and he will start his speech now.
Yeah, hello everybody. I'm just going to, yeah, brilliant. Sorry, yeah, hello everybody. Yeah, as Willie said, my name is Michael Whelan, and I'm just going to present a bit about the Buki encapsulator, which the technology which they call Prilling by vibration. Just before I start, I'll just quickly explain a little bit about myself. I've worked on this Prilling by vibration technology for about 13 or 14 years now, from my PhD to research with companies, to actually then launching products on the market. So I've seen the whole spectrum from the lab all the way right through to release of products on the market. And of course, I've also been involved with different design developments of machines, etc. And I've done this across the pharma, food, and biotech sectors. I have previously worked with Buki in the past. I think that's important to say, for about 2 years, but now I work for different companies in the technology itself. I guess I hope with this presentation for about 45 minutes is to give you some of the background of the technology and, more importantly, some of the applications within the food, pharma and biotech sectors themselves. So just, I suppose I start off with what a definition of microencapsulation is, and really it's just as defined as a process which involves the envelopment of pre-selected core materials when they're defined pores or in terms of a membrane. So you just pick your solid liquid or gas and you encapsulate it within a shell. At the bottom there, there's probably, you could say, a little bit of, sorry, do I have a technical issue? Yeah, so usually, normally, when it comes to size, we say that microcapsules are between 1 and 1,000 microns. But you'll also find a literature, you know, it can be anything from 300 nanometers up to several millimeters in size. So for this presentation, I'll term microcapsules as anything within that 300 nanometers up to 4 or 5 millimeters in size. But of course, technically they could be called macro or nano. So Alan will actually present then later on a bit about spray drying. So spray drying really is, as Alan will say, was actually the conversion of is the production of a powder from solutions, emulsions, suspensions, etc. It is important to point out as well, though, is that spray drying can also be used as encapsulation, where you actually entrap something within a matrix itself. So you can actually entrap a drug or a bioactive within the powder particle itself, and that can be termed as encapsulation. For anybody who's on the line here that's a pharmacist or is involved in the pharma industry, the word encapsulation will be probably a little bit misleading in this presentation. I mean, most pharmacists will associate encapsulation with a hard fill or soft filled capsules themselves, where such as you use fish oils or etc., which we have in the picture here on my right. For this presentation, microencapsulation will really refer to actually making of these particles in the middle with something entrapped inside. So it won't refer to the classic definition in the pharma industry where you're making a hard liquid gel capsule or soft gel itself. So it's actually going to be talking about production of particles with something entrapped inside, and that's what we refer to as microencapsulation. So just quickly mention about Buki's technology, what they have. I mean, Alan will probably mention a little bit later about their spray drying technology, and I'll talk on the right here for the technology, what they classify as prilling by vibration. And along the bottom there, it just gives you an example of the particle sizes that the machines can make. So you're talking anything from 80 microns up to 2 millimeters and beyond. It is important to point out that that's not the size range you make in one go. It means the size range you're allowed to make. So you can actually use the machine to make particles that are 150 micron in size, and they will be a standard size right across that range. Then you can change the nozzle setup and make ones that are 2 millimeters in size. Also, this here actually also talks about wet particles. So the Buki Prilling by vibration machine makes wet particles. They normally come out wet, not like the spray drying. So for anyone who's in industry or who'd like to look at getting a product on the market, it's important consideration afterwards what you think you need to do to dry them. So the particles in general will come out wet. So we have actually a picture at the bottom here just showing some of the particles that you can make from the machine itself. So this is just a standard fish oil, which we encapsulated with an alginate membrane. Sorry, an alginate membrane with the oil inside, which is coloured. So why use this machine? What's the benefits of it itself? Well, as already mentioned, it can make particles from say 80 microns up to 5 millimeters in size, with the possibility then afterwards that you have to dry them. They are spherical and homogeneous particles, equal size and shape itself. And just again, I just mentioned that already about the encapsulator, so it's quite an ambiguous term. So Buki actually referred to this technique as a vibrating nozzle or Prilling by vibration. Prilling meaning the production of small particles by breaking up of the liquid, and they do so by vibrational frequency. And I'll just explain how it works afterwards. But here we just on the right we have examples of some of the particles that can be made using the technology. So at the top again we have this core shell capsule with a liquid inside, or we can entrap a liquid directly inside a bead matrix itself. Down the bottom it's quite a thin layered capsule itself, so you can actually control how thick the membrane is. So that can be important for controlled release purposes. And along the bottom here we have dry particles. So this is where particles that were produced by the machine, and then we subsequently dried them as well using something like a rotary evaporator or something like that. So it is possible to do that as well. It's quite an important consideration, say from an industry perspective, if you want to store them for long term or for shipping reasons, etc. So how does the technology work? Well, it's quite a simple enough operation. If we look over at the side here, number one, we make up our polymer membrane, and we can mix the material that we want to encapsulate inside this. We then pump it through to number 3 there to a vibrator. And what that does, it actually vibrates the liquid itself, and as it passes out through the nozzle, here it's vibrated and it breaks the jet up into equally sized droplets. And then these droplets then land in a solution where they're gelified into a particle itself. What's I suppose the mechanism of gelation or how it's formed depends on the polymer you use. A common example of the material used is alginate, so that's based on ionic gelation. So you have a calcium alginate, which is mixed with calcium chloride, so that actually forms the gel structure. But you can also use things like gelatin, which are based on heat. So the main goal of the vibrator around number 4 is really to break up that polymer into equally sized droplets, and then afterwards they harden by different methods, and then they give you this perfectly shaped homogeneous beads, which of course you can vary the size by changing the nozzle setup. And here's just some real-time pictures of it. So we have here is just the pumping of the liquid through the nozzle at the top, and we get our perfectly formed droplets. What happens here is that you can actually cause the jet to deflect so that the particles don't hit each other, and this allows you to keep the homogeneous shape and size. Of course, then they land in a bath where they harden or they gelify, and here's our perfectly sized and shaped beads afterwards. Just on the negative charge, it can be something that allows you to get a size deviation of less than 1%. So it's quite important if you have something like cells or very expensive bioactive, which you don't want to waste much of the product. You can have very, very little waste, and all the particles will be the same size, and usually the load of the material will be very similar. Just regarding the droplet formation, there is different setups in the machine itself. So you can have this matrix structure. If you see at the bottom, we can have empty beads, or you can mix the polymer with an oil and extrude it together, pretty much like a spray dryer does. But if we look at the second set of pictures, we have this what's called a capsule formation. So with this technology, you can form capsules. So that's where a defined shell around a defined core in one step. So instead of just extruding one material through the nozzle, you extrude two, an inner and an outer. So this is quite a convenient process because it allows you to get the capsules in one step. In a lot of cases where you look to make beads, you make the core bead first with the material, and then you carry different, you can perform different steps like Worcester coating to coat it to make this capsule formation. But with the Buki machine, it is possible to make these core shell capsules in one step. And again, we just at the bottom here we have this we have an oil here with a drug inside, which has an alginate shell on the outside. And the great thing about it as well is that you can control the thickness of this membrane on the outside by using different setups on the machine, so which is quite important for controlled release purposes. If you look out there as well, several companies now also have a triple nozzle systems where you can put 2 layers on as well instead of 1, again for controlled release purposes. Or it could be the fact that you want the material itself, which you're encapsulating, to survive the stomach, so you put it 1 layer on the outside, which allows it to survive the stomach, and then it goes into the intestine and breaks down. So you have all these options available. So just some of the benefits again. So the capsule characteristics: they're small and spherically shaped. They're individual structures and they're homogeneous with a narrow size distribution. You can get as low as 1%. Normally, you would work on the size distribution of about 5%, and that includes scaling up to industry as well. It's quite a great technology and very reproducible, and it will allow you to get this lower size deviation. Characteristics of the technique itself: there is a short production time under mild conditions. The process itself of making the beads or the droplets is vibration driven. So it's simply pumping it through nozzles, and then you just vibrate it, and then there's a reaction afterwards. So it is very, very mild conditions. Unlike something like spray drying or maybe a fluidized bed or other technologies where they can use heat, this technique doesn't use heat, of course, unless you want to use heat. It's really important in mild conditions is because it can be used to encapsulate cells, whether they're animal cells, stem cells, bacteria, etc., and it doesn't cause the cell death due to harsh conditions. It's controllable. It's relatively simple and low cost. High efficiency, as I said, there's going to be very little waste in it, including when you scale it up to large scale. It can produce a range of different sizes, so you're not limited to one size range. You can anything from 80 microns up to several millimeters. It's repeatable, very important for the pharma and the biotech industry. Normally once you set your parameters here and you go to produce it again, you usually you always get the same characteristics of the final product. Also with dry particles, it does make the particles wet, but there is techniques available to dry the particles afterwards. And Buki themselves can talk to you about multi-nozzle systems. I know they have up to 6 nozzle systems, but they do work with industrial partners who can help scale it up to larger quantities, where you're talking tons a day or even more. So there is that possibility out there now. So Buki themselves have 2 systems that they operate. So that's the Encapsulator B390 and the Encapsulator B35 Pro. The one on the right, the Pro, mainly it would be a more spec'd up model because it can be used in the pharma and biotech because it's a fully sterile process. So it can encapsulate whether cells or drugs under a completely sterile environment, and we'll talk about that later. So that's the main upgrade with that machine. So this table just quickly talks about some of the characteristics of the 2 machines themselves, whether it's the size. I know we've mentioned already that it's 80 microns, so there is an adaptation you can make to the machine that allows you to go as low as 80 microns and again go higher than the 2 millimeters that is specified here. Both machines work with a single concentric nozzle. Hot prilling, what it refers there is that the Encapsulator B90 allows you to heat up the solution that you want to make into beads. So this is quite important when you're using materials like gelatin. So gelatin itself will have to be heated up to about 60 degrees beforehand. So the Encapsulator B390 allows you to do that. That function isn't available on the Pro. So it is quite important that you talk to the guys beforehand and outline the exact uses of the machine itself. There is a syringe pump on one of the machines, and that allows you to get a very accurate flow if you're looking to encapsulate something like cells. And again, as I mentioned, there's the sterile work. So the Encapsulator B395 can be used under fully sterile conditions. The 390 can actually fit underneath a laminar flow, so you can use it aseptically. And we have worked in projects before where they have used it and shown that it can offer a complete sterile environment if it's placed under a sterile hood. It doesn't have much of an effect on the laminar flow within the hood because it's quite a small machines itself. So more importantly, I suppose, for most people, is the application of the technology itself and what it can do. So I'm going to try to go through some exact applications that I've worked on over the years with the technology itself, both in the food, pharma, and biotech industry. So just going to briefly outline quickly reasons why you would encapsulate. So there's 5 main reasons why usually you look to encapsulate a material within a particle itself. It could be protection of the encapsulant, whether it's a drug, a cell, a bioactive from environmental interactions, or masking flavors. And masking flavors is quite a big area within the food industry itself, where you maybe have an oil that has an undesirable flavor and you want to mask it itself. So just simply put, you put it inside a matrix and you protect it from temperature, light, or O2. You could have a sustained, controlled, or time release of a material. So you have a drug, etc., that you want to release, whatever, say within the stomach or the intestine, and you can actually do that. So you can actually have this kind of sustained or controlled release, depending on the polymer itself and the capsule material. So you can actually control the release of the drug within the intestine or maybe the stomach itself, or you want to target specific compounds. So you can again look at making the bees themselves with certain materials where you can target along say the good itself, or you can even use them for things like foods, where you can make them you can put a bioactive in a food, and you can the bioactive itself won't react with the food straight away. It's in with a capsule, and then as when the person eats, the mechanical action of their teeth can break open and release the material itself. So you can also have something like extraction aids. In this case here, we just talk about where you can use them to extract materials from water or pollutants, or drugs themselves within the pharma industry. So they have been used in the past where you make these particles, you can put an extraction aid inside them, whether that's a liquid or a solid material, and you control them into a process like a manufacture of an antibiotic, and they will absorb the antibiotic up during production, which allows a pre-purification step, and then you can recover them afterwards, and it reduces the downstream processing. You can do the opposite as well. You can use them. You can use them the opposite then as well for something like delivering media components within a fermentation environment itself. So you can put some media within them, and they can actually slowly release that media as well. And then there's the one I already mentioned already, is convert liquids to solids. So if you have any type of liquid which you want to add to a food or a process, even something like brewing, where you can have a liquid hop and you don't want to add it directly to the process, which can affect the fermentation, you can encapsulate it inside an analgen or gelatin or a biomaterial, and it can actually slowly release, or you can break down so it can release at a particular type of time. So here's just some of the examples of materials that have been encapsulated using this technology, and I'll go through some of them now. So you have everything from bioactives to functional foods, vaccines, enzymes. I mean, it's quite important with the vaccines and the animal stem cells is that it's a gentle technique, and it allows you to do that. And across here we just have encapsulation of some CHO cells themselves. But it's not just limited to this. There's even likes of tobacco products as well, hydrophobic essential oils, pesticides and herbicides as well. It has such a wide application because it allows you to make these particles of a controllable size and shape, so you know exactly what's inside each particle. And from a delivery perspective, whether it's a drug or a herbicide or pesticide, this is actually great because you know that each particle itself it is going to be of a very homogeneous size and shape, and you can then predict what the release of this payload will be. So we just talk about quickly about the food and beverage industry. So encapsulation of bioactive materials themselves. I just read the statement before that, you know, the 21st century customers are demanding miracle foods, and whether they're safe, nutritious, economical, convenient, great tasting, and also enhance health. And it's quite a challenge for companies now to achieve this goal of getting these miracle foods or that we see more and more on the market. A lot of them are trying to achieve that by incorporation of bioactive ingredients into foods themselves, whether it's a fish oil or vitamin or iron or etc. But a lot of these bioactive ingredients are quite problematic to put inside food, usually because they're unstable, they react. They can also have an effect on the organoleptic properties, which can affect the smell and the taste, etc. So a lot of them have been and now look at encapsulation, where you put the material itself, the bioactive, into a microcapsule or a particle to allow you to deliver to the food to release its benefit, but it doesn't affect either the drug or the bioactive inside it, or affect the food. So I mean, a common example might be a fish oil, for example, which can get rancid if you put it inside food, but it is possible to put it inside these particles. It doesn't go rancid, it doesn't affect the food, and then when the customer goes to use it, then they still get the benefit of that oil itself. So I mean, the main point, I suppose, is that they're actually using the technology to overcome these problems of being unstable, reactive, etc. It is a cost-effective delivery system, particularly now that you're able to scale it up, and it has more and more polymers that you can use. And so far, the food industry has been the biggest benefactor of the technology itself. So here's just an example of encapsulation of probiotics. So for anyone that works in the field, probiotics can be quite challenging to deliver because they can break down within the harsh conditions of the intestine themselves. Processing conditions as well, putting them into the food, can have an effect on them. So the goal here is this is actually a particle here we see on the left, is to encapsulate the probiotic itself within the microbead to protect it from the manufacturing conditions that you put into the food, and then allow it to survive the stomach and then be delivered in the intestine itself. So this is work that was published in International Dairy Journal by Sinead Doherty, who actually subsequently went up to set up her own company called Anna Bio, which works a lot with the technology. What the paper discusses is how she encapsulated this strain of probiotics within a bead structure, and it allowed it to survive the pH of the stomach and pass on to the intestine, where and uptaken by the body. So the first graph here just shows that when she encapsulated the top line, when she encapsulated it within a bead structure, it completely survived the stomach over a 2 to 3 hour period. The second line is when she just mixed the probiotic with the matrix material she used for the bead, and it didn't have much survival, which shows that the structure of a microbead itself, it's not just the material that's there that's making the structure, but also the microbead structure adds to the survivability of the probiotic. And the bottom one here is just to show us that the cells themselves, if you add them directly to gastric conditions, they'll just all die over a 30-minute period. And the second graph just shows that it's a controlled delivery, because we had within the intestine then afterwards, after it passed through the stomach, it went into the intestine, where you had full release of the probiotic itself. So it's quite interesting research. So it just shows that you can encapsulate the sensitive materials like probiotics, etc., within a bead structure, and then you can survive conditions like the stomach, and then you can have a controlled delivery of them as well. So as we already mentioned, fish oils, which is quite an important one. So it's really the improvement of our organoleptic properties, whether it's taste, sight, smell, or feel, and it's probably the biggest application of the technology. So instead of adding the oil directly to the food, it is possible then to put it within either a microbead structure, kind of a matrix structure, or you can have this kind of core shell. The core shell then, it can allow this burst release as well, so you can have a burst of flavor, and it's commonly used now with companies like Pepsi and Coca-Cola, etc. So instead of the oil, or maybe it's a flavor component, instead of adding it directly into the drink, you can put it inside these beads, and then when you open up the drink, you can either, by the mechanical action of the mouth, or you can burst release it. Or a lot of people now put these particles within the lid. So you see these click lids now, which is quite common, particularly in the sports industry, where you want to add things like iron, maybe whey protein or creatine, etc. And these can be quite sensitive, and they can break down within the liquid drink. So you can put them within the cap, within a microbead structure, and then you click the cap, and it releases into the drink and releases the flavor. So it's quite common. You'll see that now very much in the sport industry, particularly for things like creatine, which are known to break down within liquids within 30 minutes. But the main application though would be taste and smell, particularly fish oils, that you want to add to foods for their health benefits, but they can go rancid, they can react, etc. And you even see things like tea as well. It's another one where you want to add components like flavonoids, etc. But of course they can react. But encapsulation prevents this reaction, and it can release either straight away or have this burst effect. So just if you go through literature, and I mean, a lot of the work that's been done in this technology, I suppose I can't talk about what I've done in industry directly, but if you do take a look through the literature, there is several studies that talk about different oils, etc., and foods that they have used with this Bookie technology itself. Now you'll probably notice that when you're reading the technology, it'll call it either vibrating nozzle or Prilling by vibration, and you'll see that it was another company that made the technology called Inotech or Encapsulation. So it's the exact same technology. Bookie acquired those companies back in 2012. So it's the exact same technology. So you're reading through the papers and you see some of these oils, etc., that or foods that have used this technology, and it's a different name for the company that make technology, but it actually is Bookie themselves who make this machine, and it is the same company. You probably will find as well a lot about the technology is that it works brilliantly both in lab and large scale, and a lot of companies tend to take in the technology, use it to make a product, and they don't talk so much about it afterwards. So it can be quite hard to find some literature on it from companies particularly, because they find that it gives them a competitive advantage, because it allows them to make microbeads or capsules that they want and put new products to the market. So you will find a lot of literature on it from universities, but from companies they tend to be a little bit more quieter because this technology gives them such a competitive advantage that they don't like to discuss it. So other applications of the technology, well, you can have delivery flavors, whether they're volatile, which I already mentioned, say for soft drinks, etc. You'll see in patents from PepsiCo, not necessarily using this technology as well. There can be other technologies that allow you to produce similar microbeads. You could have things like visual effects. This is quite big for the kids market. So you can have beads or microcapsules that look good, and you can incorporate a health product inside them, and kids visually they look great, especially for kids. And alcoholic drinks there I have as well, because you can see now more and more companies are using them for cocktails, etc. They're more aesthetic looking, so they can put particularly the gin industry, for example, you can put different flavors inside them, and they can have a slow release of a flavor in the cocktail, for example, and it looks good too, and people like it. I think bubble tea, which initially started a few years ago, had to do something similar as well. Again, the stabilization and production of nutraceuticals. So this could be vitamins and minerals. So this could be things like iron or flavonoids, which are very unstable. They can react within the food, and these reactions can render them non-bioavailable or not good, or they don't have their therapeutic effect anymore. So you can encapsulate them within a bead structure itself, which protects it, and then when it gets when the customer takes it, it can be delivered whether it's the stomach or the intestine, etc. So I even some iron. I encapsulated some iron within gelatin beads to protect it within the iron 2 state. So just because I was just talking about the food industry, and just mention quickly about molecular gastronomy, which is now also another area which is quite keen to use encapsulation technology in general. So you have people like Harvey Teese or Heston Blumenthal, which people would actually see a lot, and you'll see some of them in the fancy restaurants now in London, or you might even see it on TV. I see it a few times that you can see that they now use things like, they use similar to this technology, these micro beads, where they have encapsulated different flavors or different flavors within beads and added them to the food themselves. So they usually call them they usually call them, I forget the name they have on them now. They usually call them like caviar beads or something. So I suppose the point here is it just proves that the right range of applications that technology itself has. If we're talking about the pharma industry itself, so there's a lot of work going on the production of active ingredients, with the main goal of reducing interactions with other agents during storage or during transport through the stomach. So there are two main reasons you would use it. So you have I suppose a very sensitive and very expensive API. So this technology allows it to, could be for long-term storage, or mainly though would be delivery in the intestine. So you want it to pass through the stomach itself and then into the intestine. And the great thing about this technology is the beads themselves are very uniform. So you can actually encapsulate your active ingredient within a micro bead, which are all similar size and shape. So it's very, very important for controlling dose. So you know that you add 50 beads, you know that the exact, close enough to the exact amount of drug that they can add. Another good advantage within the pharma industry is that the particles produced, as we see in the second picture here, is that they are very, very uniform, so it allows coding. So if you want to code it with a Worcester process, it's quite an easy process to do that. So if you want to add additional layers or coats, whether to pass for passing through the stomach, etc., or for storage reasons, or to protect it from oxygen during storage, you can do this quite easy with this technology, because it allows you to produce very uniform beads, which are spherical and have a smooth surface. So when you go to use it in a Worcester coder, it's quite a simpler process compared to maybe if you're using powders themselves. What we have here is PLGA particles, which have a drug inside, which are an anti-inflammatory inside themselves, and then these would have been passed on to code it, and they were coded at quite a high efficiency. So the goal really was, the goal of it was to protect the active inside from oxidation during storage, and that breaks down in the stomach, and then the material that's left is the PLGA, and then that breaks down and releases its payload. So this is just an example of a company as well who I've seen on the market have used our technology, and then what they do is this is the combi caps where they make the micro beads, and then they can actually fill them within or encapsulate them within a hard or soft gel capsule. And there's many different types of application here. So you can make your bead inside here with your drug, and then you can actually maybe fill it with an oil itself, or fill it with another material. So you can have combination products. So maybe you have an oil here that that allows your drug to be solubilized better in the stomach, etc. So you can have your oil here, and you have your microparticle in size, and then it allows better delivery of your active ingredient that you want to deliver. So again, these are just so many examples in literature for anybody who's interested in looking them up of some materials like furosemide, thalidomide, etc., that use the technology itself, and they're available to, and they actually just show you the polymers, etc., that they use. So I just want to mention now a bit about the biotech and transplantation, or biotech and medical applications as well. So the technology itself, one of the main reasons for its development back in the 80s was when this vibrating technology was first developed, was for medical applications, mainly encapsulation of cells for human transplantation, particularly islet cells for diabetes. And it is still an area which gets widespread use of the technology, and there are several companies who've gone into clinical trials with the technology itself, say for islets, or even things like stem cells for the treatment of Parkinson's disease, or dopamine producing cells, etc. There's companies in say Australia and New Zealand are in a phase, I think a phase 2A trial using this kind of technology to deliver dopamine producing cells to for Parkinson's disease. So the whole goal is that you can encapsulate the cells. Say we have islets within a membrane, within an alginate membrane, and then you can transplant them inside the body. Because the cells themselves are inside this matrix, the body itself can't attack them. So the cells can actually proliferate and grow and produce their therapeutic products. But there is a porous structure within the membrane, and it allows say the food or nutrients to come in and waste to go out, so the cells can grow and then produce its product. So I actually have a video here I want to show, because the Buki machine has been used, as said, by several companies and universities to transplant cells. So this is a BBC video of University in London, which used the Buki machine to encapsulate cells and then transplanted them into a kid to help them overcome the liver disease. So there was actually 5 kids a part of this trial, and the process actually helped the kid overcome this liver disease. And you'll see the machine itself being used in the video, and it'll explain how the technology itself works. So I hope everybody can see this now. I'm just going to play the video.
Meet a medical marvel. It's hard to imagine now, but 6 months ago, Ayad was close to death. A virus was destroying his liver. Now it's working normally. His parents say their only child has been given back to them.
It was great. It was great. Once he had the treatment, immediately after 48 hours, things started slowly to get better, and our hope come back. We are very proud of him. He's brilliant. He's a miracle boy.
What saved his life was not a transplant, but deep frozen human liver cells. Scientists at King's College Hospital
So just in the picture there, actually,
coated the cells with a chemical.
the machine here that's just shown is actually the Buki machine itself. It's an older model of the machine.
Found in algae to prevent Ayad's body from rejecting them. You can see the coated cells at the bottom of this jar. Instead of going on a waiting list for a transplant, Ayad was given a single injection of liver cells. Their protective coating was porous. This allowed toxins to flow in, be processed, and waste products and vital proteins to flow out. Immune cells were too big to enter, so couldn't destroy the donor tissue. After 2 weeks, his liver had started to recover.
The left lobe looks normal.
A key benefit over a liver transplant is that Ayad will never need anti-rejection drugs, immunosuppressants. Doctors are delighted.
It's only a few months back when I first saw this child who was so sick, requiring support to dialysis on a breathing machine, and we think that we have given him another chance of life. And seeing him now 6 months down the road with nearly normal liver, without a liver transplant, is remarkable.
But doctors are urging caution. This world first might be a one-off, so a large clinical...
Yeah, so yeah, so I just think it's an important video to show that the kind of applications that can be used with the technology. It's not just something that's predominantly used in the lab, but it has real world applications both in the pharma and the medical sector. And as I said, there is several trials going worldwide, looking at not just liver disease, but there's also diabetes, which is a huge area for it. Encapsulation of stem cells for treatment of things like Parkinson's disease, and also the treatment of Alzheimer's disease. And the important aspect to mention is that the technology is so gentle that it doesn't damage the cells. They remain viable afterwards. It's fully sterile. It can be used in the lab. It can be used at very small quantities of cells. So in many cases, when you're harvesting these cells, you have very small amounts. With the machine itself, you probably have only a loss of maybe a couple of mils of the solution at max. So all of these reasons allows it to be used as a machine that can encapsulate cells for human transplantation. Again, just some of the literature again. If there's some available literature available again for different things like stem cells and therapeutic proteins, vaccines as well is something that's come very big with it as well, because it is a gentle technology and it's controllable size and shape. And even at the top there, another thing is even within the bovine industry as well for things like sperm encapsulation, etc. It's become more and more prevalent with the technology again, because it's gentle and you can use small amounts. And in essence, the machine, the lab scale ones are quite small and nimble, and can be brought to different locations. So just something about encapsulation of CHO cells. So for anyone who works in drug production of therapeutic proteins in large scale in industry, will know that there can be a lot of challenges here, mainly because of low productivity of the final drug itself due to hostile environment. So if you're using a CHO cell line to produce your therapeutic protein, as we know, they don't have cell walls like bacteria or yeast. So when you put them into your fermenter, they don't proliferate as much, or they can be damaged, or in many cases they can die. This reduces the low productivity of the drugs, which significantly increases the overall cost of the bioprocess itself. And in many cases, the production is not economically viable. So you have cases where you have a recombinant protein itself, which is clinically quite important, but it can be the case where it doesn't, you can't produce it at large quantities to make it economically viable. So you have overly complex techniques to overcome these problems, like perfusion cultures, etc. So what we looked at in the past as well with several companies is that we encapsulate the cells themselves. These red here are supposed to mimic the cells. So you encapsulate the cells within a core shell structure, and then you actually add them directly to the bioreactor. And we call these kind of a microcapsular structure. So you have cells encapsulated with a membrane, and you add them to a fermenter. And what it allows is that the cells themselves, as we already mentioned before, this capsule structure is semi-permeable, so it doesn't allow, it allows nutrients and waste out, and the bioproducts to be produced, but it protects it from the harsh fermentation environment, such as shear stresses and forces. And so what we've seen is then that it allows the cells to grow to much higher densities than you would get in a normal bioreactor. Hence, it actually produces more product, or more bioactive itself. Another, I suppose, advantage as well is that what we looked at in the past as well is that you can actually use this cell as well to accumulate the bioproduct itself. So instead of it passing back out into the media, and then you have to purify the product using complex downstream processing, which can account for about 80% of the cost, you can allow the product itself to build up in this capsule. So at the end, you just have to remove the capsule and break it open, and you have a pre-purification step. So it can significantly reduce cost that way as well. This is just some examples, some work we performed at DCU using this within a bioreactor. So what we have here is in the clear boxes is when we encapsulate the cells and add them to the bioreactor, and in the blue here in the circles when we didn't. So you can see that there was a significant increase in growth of the cells. So there's a fourfold increase in cell density, and the growth rate was increased by 1.6-fold. So they actually form. So here's under a microscope we have the cells encapsulated, and this is the growth of them over 8 days. So you can see them actually growing inside. while they're in the bioreactor. And I said there was actually a fourfold increase in cell density, and the growth rate increased by 1.64, which can be massive in large scale when it comes to economics of a process itself. And this just actually shows the production of the IgG which was produced. So there was actually not just the increase in growth, but there was an increase in 15.5% in the product itself compared to the suspension culture. Even though there was an increase in cell density by fourfold, so 400%, there was only an increase of IgG by 15.5%. And many people probably ask why it wasn't 400% if the cell number had gone up so much. And what we found out was that the cells were growing so quickly that they were using all the nutrients to grow, opposed to actually just produce the IgG1. So it's something that the research wasn't continued on further, but in that case, you would just increase the media load, and hopefully the more product would be produced. But it is just an example of one of the uses of the technology itself. I'm quickly just going to talk a bit then about extraction aids. So I mentioned already that when you're looking at recovery of compounds, so so far we've been kind of talking about keeping materials inside the capsules itself. But here's just an example of where you can actually use them as extraction aids. So you can put a material inside the beads, and then you can add them to a fermentation, or you can add them even to a wastewater treatment, and they can actually absorb material, whether it's a pharmaceutical within a wastewater treatment plant. So in a lot of cases, wastewater treatment plants are not designed to remove pharmaceuticals, etc., which are building up in the environment. Mainly due to overuse, and so the normal case is that they're not always broken down by the body. They pass into the sewage system, and then they're actually bioaccumulating. So what we found out in the past is that we could actually make these micro beads or particles, and we could add them to wastewater, and they would actually absorb some of the commonly found drugs that are in the water, environmental waters. So they can actually absorb them, and you can put a material inside them that was very, 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 very efficient at taking out these drugs. So this is and I suppose one of the points here people might think is that you're adding microbeads or microcapsules in the environment, and we see that there's a lot of problems with that at the moment with cosmetic industry. And I think it's important to point out that these are made from natural materials, so they do actually biodegrade over a period of time. They're not made of plastics. But it is possible to remove them quite easily, and I mean that was the goal of the research, because after they recover the drug, you don't want to leave them in the water. And what we found out is that we can actually control whether they sink or float. So we can put materials inside them, whether they will actually float to the bottom where they can be collected, or they can actually float to the top, or they sediment to the bottom, and they float to the top where they can be collected. You can put them inside mesh filters or mesh bags, or you can even put magnetic particles inside them, where you can then collect them afterwards as well. So again, there's a huge range of different areas you can use them for, and because of the setup in the machine, and you can incorporate different kind of characteristics into the capsules themselves. So this is just example of some of the work I've done myself. So on the first graph, you have like 7 drugs that are commonly found in wastewater treatment plants, and it just shows you that this here is just actually the removal of the drug itself. So you can get from between 15 and 100% removal of the initial drug concentration within a 50 minutes period. So it's quite a rapid extraction of the drug. And it is, in all cases, the amount of the drug removed by this technique is higher compared to conventional treatment plants. And this is like a very low concentration. We only added about 1% of the beads. So to increase it for things like furosemide or that, which still have high concentration left in the water, but you just increase the number of capsules. And on the right here, we have pesticides and herbicides that are commonly found in the water. And again, we get 25 to 75% removal within 100 minutes of that pesticide. And again, it just shows how rapid the removal of it is. I suppose a lot of people might ask, what do you do afterwards with the drug? So here at the bottom, we have a pesticide inside the capsule after we removed it. And what do you do with it? And there is things like, you can incinerate them, etc. But what they found out afterwards is that normally you can have things like a Pseudomonas culture which can break down herbicides or pesticides, but normally the levels that the herbicides and pesticides are at can be quite toxic, and it causes the cells to break down. But what they've done is here to remove the bead containing the herbicide and pesticide, and what it is that they put that into a fermentation containing Pseudomonas cultures, and the bead themselves, it allows the actual the drug to slowly extract out at concentrations that won't kill the cell itself. So this is the same kind of principle to use for releasing a drug within the human body. So it releases very slowly, and the Pseudomonas can break that down. It doesn't reach its toxicity level. So then it just grows, and it keeps on going until it breaks down a lot of the drug itself, or a lot of the pesticide itself. And then afterwards, you just have to autoclave the Pseudomonas, and you can discard it then as well. So instead of having to incinerate them or use other kind of chemical process to break down the herbicide and pesticide after you remove it from the water within the capsule, you can actually carry out this process, which is quite a neat process. Just quickly there before I finish up, these are just some of the products in the market that kind of use technology, or definitely use similar techniques to this technology. So you can have everything from the top here, from coffee. So you can make coffee beads using this technology, and inside that you can incorporate bioactives or probiotics, etc. Sorry. We have chocolate balls here with probiotics again inside it, quite common for the kids market. So again, it's quite a gentle technique. So you make these coffee or these chocolate balls, which kids love, and you can actually have probiotics inside them. The chocolate balls. This is a company you'll see online, a Belgian company which uses technology for probiotics. There's underneath is again an Australian company which uses it again for probiotics as well. You can even have, if you look at your chewing gums, they also have it for mainly for flavor of release, but more and more of them are looking to get into the market of bioactives, or even sometimes some of them are looking actually for to put inside the particles something that will prevent the chewing gum sticking so much, or could help with its removal afterwards from pavements, etc. You'll see a litany of products, cosmetics, and I don't want to go into it too much, that are using these microbeads as well for things like flavors or to help in the cleaning process. There is, I suppose, a lot of negativity around this now with the build-up of these microbeads, but the Buki machine itself, it does allow. I mean, a lot of these materials we're using are plastics. They really are biodegradable materials, so you can still get this release of your cosmetic or your active ingredient within your cosmetic, and these then will actually break down naturally in the environment itself. I'm just wary of time. I'm going to skip on with a few slides. These just show you some of the other applications. It's kind of going away from what we talk about. But I just want to mention a little bit about the matrix materials, so the material that you use on the machine itself. I mean, these are the more common ones that are used at the moment, from alginates to whey proteins to waxes, etc. It's quite an important consideration here if anyone's looking to scale up the material you use here, because in many cases, such as gelatins or carrageens, they're natural polymers. There can be a variation between batches that you get from suppliers. And another important thing is, remember, and I mentioned earlier, is that when you use these materials, they're usually at a concentration of, they're usually hydrogels at a concentration of between 2 and 5%. So after you made them, you have your microbead, and it has probably a solid content of 5%. They then have to be dried. So that's an important consideration there, that to dry a particle down from 5% solid to 95% loss of water. It can have huge issues when you go to producing 1 ton or 10 ton of the product itself, because in essence, if you're producing 100 kilos and you only have a 5% solids, you're losing 95% of that, and you're only getting 5 kilos. So in industry, a lot of the time they use gelatin, because gelatin you can use at high concentrations with the technology, so you're 30–40% solid content, so you only have to lose then 50% water. So it is an important consideration to think about, and what a lot of people do is that they put fillers in, etc. But you have to examine how this can affect the production itself. So it is important for anyone. I mean, in the lab itself, it's not really an issue when you're looking at a new applications. But for anyone that wants to go to industry, then the metric material is quite huge. Not just from the fact that it's batch-to-batch variation and the supply of it, but certain materials won't allow you to have a very high solid content, and you need to dry it afterwards. I think the bookie can talk afterwards about they do have a multi-nozzle system as well. It is available, a 6-nozzle system. And I'm just going to quickly, 2 minutes mention, and I'm going to wrap it up. There is companies that use it at industrial scale, and you can have, you know, I've worked on processes myself where you have 80 to 100 nozzles. And there's even higher where you have 1,000 nozzle systems, and just actually a picture here. It's not a very good picture, apologies. So normally the bookie system just has a 1 nozzle, but you can have these systems with plates where you can push through tons of product in a day itself. So there is a lot of companies. There's not a lot. Sorry, there's several companies that do that now on a large scale, and I'm sure bookie can pass you on to them. So for anyone that's here from industry who's looking to look to getting the product at a large scale, there is a possibility there. There is a lot of available information on the bookie website about the technology itself, and there is upgrades as well. I'm gonna just cut through all of that, and the take-home message quickly is just that it's, you know, the technology itself has the capability to produce from 80 microns up to 4 or 5 millimeters inside. You can mainly produce wet particles, but they can be dried afterwards by things like maybe fluidized bed, etc. You can use it for hydrophobic or hydrophilic material. It can be sensitive or robust ingredients. Particularly the sensitive one is quite important. It can be organic or aqueous. And there is an endless number of products and processes. I just haven't got time to go through everything today. And it can vary right across the spectrum, from food, chemical, pharma, cosmetic, tobacco, herbicides, pesticides, environmental, any area that's looking to make repeatable small particles under controllable characteristics for large scale. I mean, this actually ticks all the boxes. For myself, I've seen massive improvements in the technology, particularly in large scale, where it's now you're able to produce quantities that are needed for an industrial level. Previously, in the past, it hasn't been the case, where it's mainly used in the lab, but now we see more and more companies come on board with it, so you can actually scale it up. And just for anyone that's in industry, or anyone that's actually in academia, there is actually societies out there that share a lot of research. So there's one called the Bioencapsulation Research Group, led by a great guy called Dennis Ponce, who's been involved in this microencapsulation field for about 30 years. So they have several industrial symposiums every year, conferences, industrial collaborations, etc. And there's about 8,000 people who are linked to the organization, and it's something that you should look at joining if you want to increase your knowledge in the area. So I'm just going to finish with that. So thanks for everybody. It's a bit rushed, and but I'm going to actually just leave it there for the moment.
Okay, Michael, thank you very much. I would like to give now the word to Mr. Michael Kerwick.
Thanks very much, Willie. Sorry about the earlier technical difficulties. I had a problem with my speaker, but I'd like to thank Michael for sharing his experience. It was, as usual, very interesting presentation. Thanks for that, Michael. Just a bit about myself. Michael Kerwick's my name. I'm the business unit manager at Mason Technology. We're based in South Circular Road in Dublin, family-owned business, and we're a laboratory and industrial instrumentation supplier. We actually have been official bookie distributors for over 20 years. And at Mason Technology, we employ over 80 people, and we're a sales and service company. In fact, all our bookie engineers are factory trained. We also represent some of the world's major equipment manufacturers, such as Mettler Toledo, Shimadzu, Olympus, as well as bookie. So at this stage, I'd like to introduce you to Alan for the second presentation. Alan hopefully is online there, and he's based in Trinity College in Dublin, and he's going to talk about the use of spray drying to crystallize materials. And Alan is actually from the School of Pharmacy and Pharmaceutical Sciences. So I don't know if you can hear me there, Alan.
Yeah, can you hear you?
Yeah. Okay. There we go.
Okay, so hopefully everyone can see the screen there. Thanks very much for the introduction. Like I said, my name is Alan McDonough. I am a final year PhD student in the School of Pharmacy and Pharmaceutical Sciences in Trinity College Dublin. What I'm interested in is the investigation of the crystallization of active pharmaceutical ingredients onto excipient surfaces using a spray dryer under the supervision of Associate Professor Lydia Tajbir. I would like to talk to you today about the use of spray drying and to crystallize materials through evaporation crystallization within a spray dryer. I will discuss a general overview of the process, the transfer of crystallization theory from a conventional crystallizer to that of a spray dried droplet, the use of models to characterize these droplets to allow for greater control over the crystallization process, and the role of spray drying parameters on the resulting crystallized material. So I'd like to start with what we think about with regards to a conventional crystallizer. What we could have is a cooling crystallizer that imparts supersaturation by cooling the undersaturated solution to cross the solubility curve, or using anti-solvent precipitation, which reduces the solubility of the solute in the solvent, which you can see there. But in relation to spray drying, the method of imparting supersaturation is via solvent evaporation. For cooling crystallization, as the temperature decreases, the solubility of the solute in the solvent also decreases, up to the point at which nucleation would occur spontaneously to equilibrate the chemical potential of the system. In the case of evaporative or anti-solvent crystallization, the increase in solute concentration as a result of a decreasing concentration of solvent results in the formation of this driving force. So here in Trinity, we have two Buki P290s, which are lab-scale spray dryers. So spray drying in itself is a very versatile technique capable of drying an inlet feed of various compositions ranging from suspensions to slurries to solutions. During a spray drying process in which the material to be dried is in solution, the inlet feed would enter the atomizer at the top of the drying chamber. You can see it coming through in this direction. And then it would come in contact with an atomizing gas, usually air or nitrogen. So you can see beside the inlet feed here. So this sudden increase in velocity causes the feed to become atomized, forming droplets usually on the micrometer scale. These droplets then come into contact with the drying gas set to the inlet temperature of the spray dryer. With the tiny size of these atomized droplets, there is a large surface area for heat transfer, and evaporation of the droplet would begin. As this continues, the droplet decreases in size along with the volume of the solvent, and therefore the concentration of the solute would increase. This continues up until a point to where the kinetic barrier to nucleation is crossed and tiny crystallites would start to form. With further droplet shrinkage, these crystallites increase in size as dissolved molecules diffuse to the crystal surface. As well as this, the growing crystallites themselves diffuse towards the center of the droplet to escape the approaching drying boundary layer. So you can see it here. This continues until diffusion is no longer favorable and a particle consisting of an agglomeration of semi-spherical crystallites is formed. So this can be seen in some of the work we've done here in Trinity College Dublin, in which the individual crystallites of paracetamol crystallize onto the surface of alpha-lactose monohydrate can be seen. So we can see the lactose here, which would form these triangular shapes when they crystallize, and then paracetamol forming the semi-spherical agglomerates, crystallizing onto the surface of the lactose. What is important to note, however, is that the surface morphology of spray drying materials depends both on the spray drying parameters used, but also on the nature of the material that is being dried. Particles ranging from highly crystalline materials with defined crystal faces to amorphous materials with a hollow core and no defined shape can all be produced depending on what material is being dried, the choice of solvent, the inlet temperature and corresponding outlet temperature, the solvent mass flow rate, the atomizing gas mass flow rate, and the drying gas flow rate which is used. So here we have an image of an atomized droplet. In relation to spray drying, each individual atomized droplet is now your crystallizer. All of the process parameters that control a conventional crystallization process, as mentioned previously, still apply here. The temperature at which the energy supplied to the droplet is in equilibrium with the energy required for solvent evaporation to take place is called the wet bulb temperature. This temperature is the product to the solvent boiling point and the drying gas outlet temperature, and was first proposed by Miller et al. This is an important parameter in controlling crystallization in a spray dryer as it influences the physical properties of the overall process, such as the density, the solvent viscosity, the solvent surface tension, drying gas density, the atomizing gas density, the diffusion coefficient of the solute in the solvent, the diffusion coefficient of the solvent in the drying gas, the solvent saturated vapor pressure, as well as the solute solubility in the solvent. As a droplet evaporates and the outer wall of the droplet shrinks, the solute diffuses away from the drying boundary layer towards the center of the droplet. This diffusion is characterized by the Wilk-Chan equation for the diffusion of a solute in dilute solvent. In a conventional crystallizer, the diffusion of the solute in the solvent is governed primarily by the type and rate of the stir. However, in this case, the rate is much slower and depends on the internal fluctuations of the droplet temperature. To estimate the diffusion coefficient of the evaporated solvent away from the droplet surface and into the drying gas, Slater et al. Proposed the following correlation. It combines kinetic theory and corresponding states arguments to estimate the diffusion coefficient at low gas pressures. This then allows us to calculate the rate of solvent evaporation from the droplet surface proposed by W.H. Finley. This expression negates step and flow, however, as long as the evaporation solvent partial pressure remains small, such as scenarios where a droplet temperature is much lower than its boiling temperature, then a good estimation of the rate can be made. The ratio of the evaporation rate to the diffusion coefficient of the solute in the solvent gives the Peckley number. This is a dimensionless number for which a value of less than 1 implies that the diffusion of the solute is faster than the rate of solvent evaporation. This will show that there is adequate time for the solute to diffuse away from the drying boundary layer towards the center of the droplet, forming a spherical particle with a solid core. Alternatively, the larger the Peckley value is greater than 1, the larger the tendency for a particle to form with an outer shell and a hollow core. This is due to the diffusion of the solute being slower than the evaporation rate, resulting in an increase in surface enrichment of the solute before being caught by the drying boundary layer and forming an outer shell. Taibo et al. Proposed this following correlation with slight modification from Kemp et al. To calculate the size of droplets using the Salter mean diameter. The droplet size is an important characteristic to resolve as it determines the size and volume of the crystallization vessel. It is influenced by the physical properties of the solvent and spray drying parameters, of which the atomizing gas flow rate is the most important. As a general rule of thumb, the larger the atomizing gas flow rate, the smaller the droplet size as a result of the higher energy available for dispersion. Other factors that influence the size of the crystallization vessel is the diameter of the inner liquid feed orifice, the solvent flow rate into spray dryer, the solvent viscosity, the solvent surface tension, the solvent density, the atomizing gas density, and the velocity of the atomizing gas. From the evaporation rate and the diameter of the droplet, we can then determine the total drying time for the droplets in the spray dryer, and using the change in droplet size to calculate the change in droplet volume over time, together with the initial concentration of the drug in the droplet, the change in supersaturation can then be determined as a function of droplet drying time. So now that we have transferred the theory of crystallization using the conventional crystallizer to that of our new crystallizer, in our case the atomized droplet, we can now have a look at the effects of varying the usual process parameters in this crystallization vessel. Of course, one must be very careful in making sweeping generalizations with regards to the effects of varying process parameters across all spray-dried materials. But generally speaking, there are a few rules that you can follow to begin with to impart greater control over your crystallization process using a spray dryer. As an example, if we are interested in controlling the droplet size, as it is a contributing factor to the overall particle size, and we want to increase the size, we would reduce the aspirator flow rate. This has the effect of reducing the droplet's wet bulb temperature. As the solvent viscosity, surface tension, density, and atomizing gas density are all proportional to the temperature, they also subsequently increase. The density of the solvent and density of the atomizing gas increases to a lesser extent in comparison to the numerator, leading to an overall increase in droplet size. This effect is also seen in reducing the spray dryer inlet temperature. With a decrease in temperature leading to a decrease in the droplet wet bulb temperature, the atomizing gas has the largest influence on the droplet size and should be the first parameter considered in relation to altering the droplet size distribution. With an increase in the atomizing gas mass flow rate, there is more energy for liquid feed dispersion, leading to smaller droplets. If larger droplets are desired, a lower flow rate should be used. Keep in mind, however, that there is a limit to how large of a droplet that can be produced. If too large, in conjunction with an aspirator rate that is too high, then the spray dryer will not have the required time to evaporate the solvent before the droplet enters the cyclone and possibly the collection vessel. By increasing the solvent feed mass flow rate, there is a greater quantity of solvent and solute entering the spray dryer atomizer per unit time. This has the effect of increasing the volume of each atomized droplet and therefore its size. By using a higher concentration of water in the inlet feed compared to an organic solvent, the size of the droplet will increase. Now, it is worth noting that this parameter in particular is quite sensitive to the composition of the solvent feed and that it may behave differently with different choices in solvent, taking into account the possibility of strong solvent-water interactions. But in general, with an increase in water concentration, there is a slight increase in droplet size. This is a result of an increase in solvent mass flow rate, viscosity, and surface tension. The solvent density also increases. However, the overall increase in the numerator surpasses the increase in the denominator. Finally, in order to increase the droplet size, the concentration of solute in the feed could also be considered. This is the effect of increasing the droplet's viscosity over the increase in density, producing a larger droplet. Now, as I'm coming to the end of the allocated time, I'd better finish up. I hope you found this talk interesting and learned something new. The take-home message that I hope you have found is that spray drying is a very versatile technique, and with the right control can produce dried crystalline materials of your specifications and choosing. And thank you very much for your attention.
Alan, thank you very much for your speak. We are now setting up all the open questions which we received from the poll. Audience, if you still have questions, please write them to the question section. In parallel, I will open the poll questions which you could answer to me, to us. I will unmute Michael Whelan as well. Did I get it? Okay. Now we have to go back and forth, huh? So we go to the first question. I think this is a question for Michael Whelan. Michael?
Yes. I can't see the questions. Can you call out the question for me?
You can't. Can encapsulation technology be an application to improve solubility of insoluble ingredients?
Yes, it can. It can actually. You can encapsulate. What you can do is you can, if you have hydrophobic ingredients, you can solubilize that within an oil, and then you can actually encapsulate that within a particle. And what happens there is that it allows it. You can then add it, I suppose, to the stomach. It can break down the outer material, and because of it's solubilized within the oil can break up and then slowly release. Can slowly release actually the hydrophobic material. So it can be used to improve solubilization, yes. Normally because you can dissolve the hydrophobic material in an oil and then encapsulate that. And then you can get the microbead to break down within, say, the gastric conditions, and it releases the oil with the active inside it, and then that actually gets slowly released and improves its transport, say, or release within the stomach or intestine. There is some research on that. Sorry, I actually can't think off the top of my head.
And Michael is answering the question. It's fine. Okay, thank you very much. So we go to the next one. The next question is it possible to encapsulate only enzymes for multiple use?
For multiple use, normally what you would do with that is actually, for multiple use, you would normally make a bead and stick an enzyme on the outside of it, and it allows you to recycle it a lot better. So that's some of the work that's been done in the past. There is some literature on that. For multiple uses within the bead, inside the capsule itself, I've not been involved in any, but mainly I on the top of my head, I can't actually think of anything at the moment where you put it inside the particle, but definitely it can be used to, you make a bead and you can actually stick an enzyme on the outside of it, and then it actually is added, it controls the process, and then you can recycle it a lot quicker. So for that application, yes. If you look at online for papers from a guy called E. Morrison and Anne Wyss, her second name is spelled W-Y-S-S, they've released several publications on that type of process.
Okay. Then I'd like to go to the next one. What are the specs for the capsule intake of the pesticides?
What are the specs for the, say it again, Willie, sorry?
What are the specs for the capsule intake of the pesticides?
I can't think of them on the top of my head. Again, there's actually publications released on that. Again, Morrison and Ian Weiss, there's 2 papers. I think I might, on the presentation, have. So it's something that we can just pass on later on. But there is, the specs will be there. The whole publication is on. I'm trying to go back to the publication here. Yeah, I don't have the publication here, but yeah, there's a publication there for that. We can give you the name of it afterwards, and it'll give you all the specs that you need to know.
Okay. Then the next question is, what is the largest volume of fish oil we can encapsulate using the system with the largest nozzle concentric cap?
I'd probably have to let one of you guys answer it, because I don't know if there's been changes in the. I mean, when I was there, I suppose I mentioned earlier, I worked with the company previous. I can, from a volume perspective, it is possible probably to get loading of 80 to 90% of oil, but that's with a very thin membrane, very, very thin membrane. Normally you would have a loading of around 25 to 30% of oil within the capsule, and from my time at the company, we could make the particles between 4 or 5 millimeters in size, and as low as about 200 or 300, and they are wet particles. Just to point out that they are wet. But if you can remove the water, you can actually shrink them down even further to a smaller size. With shrinking the membrane itself, the reason you can get 90% loading is because you can remove the water from the outside membrane, and that'll get you a very, very thin membrane and can get you up to 90%.
Okay, thank you very much. Next question is, do probiotics need to be dried in order to have long shelf life when using an encapsulator?
Yeah, I can't, I haven't done enough research in this, but in generally speaking, yes. Normally, if you're gonna store something, you find that by drying it, it will have a longer shelf life, whether it's a probiotic or bioactive, etc. That's not saying that you have to dry it. You can actually add it directly within the liquid, and it will have a good shelf life. But the shelf life would significantly increase if you dry it. And it's a case by case scenario there. I can't give, each case will vary. So whether it's a say a fish oil or whether it's a probiotic, but normally what we found is that the bioactive will definitely have a much longer shelf life if it's dried compared to it it's in a wet state.
Okay, thank you very much. Next question, is encapsulation technology suitable for palm oil products?
Yeah, well, I mean, if you're going to look at it, yeah, it is for palm oil products, and we've done it before. You can either mix the palm oil with the matrix, say it's gelatin, and then extrude it and make a microbead, or you can make a coarse shell. The main thing here is the viscosity of the palm oil and what temperature you want to use it at as well. But both are possible.
Mm-hmm.
Just one thing Billy's mentioned is important is the viscosity is important consideration with the technology. So you have very, very, very viscous solutions. It can be an issue with extruding them. But you're talking stuff that's like 500-600 centipoise of viscosity, but that's very, very high. You're talking about a liquid that doesn't really flow. But there is ways around that. Normally you can actually heat it up, and that actually reduces the viscosity and things like that. So, any substance that's very viscous or is actually solid at room temperature, that's possible to use in the machine by using heat and then extruding it through.
Okay, excellent. Then next question which has been asked is, what is the best matrix for etheric oils?
For which oils?
Etheric. So these are the taste oils from, like, example, fruits, herbs, the one with or in the nose. Yeah,
so exactly. Sorry, I couldn't hear it properly. I went through that quite quickly, which was, and I probably explained it too quickly. The material itself is very important, particularly for large scale. I mean, I suppose the question could be if it's large scale or lab scale. If it's large scale, I would look to use something like a gelatin, or because gelatin has high loading. You can actually make a solution with 30% gelatin, and then when you go to dry it, you only have about, if you have solid content, you have about 40%, so you have 30% of drying to do. So in any case like that, I think gelatin will certainly be the best material. If it's in the lab and you're just using it to come up with new ideas and that, I mean, I think alginate is the best. It's the easiest to use. It's the easiest to source. It's a rapid gelation. So again, it depends on the application.
Okay. How can the porosity of the capsules be controlled?
That's a good question. There's several ways of doing that. It depends, firstly, on the matrix you pick. So if you have an alginate or a carrageen or a chylazine, a pectin, these will naturally form pores when they're made. Now, the size of that pores will depend on the makeup of that material, and the makeup of that material will depend on the source. So, for example, alginate, it depends on where you source it. It comes to seaweed. It depends on where you get the seaweed. It depends on what part of the plant to take it from, etc., etc. But the majority of them will form pores. Again, but it'll be dependent on where you source the material. You can, however, also vary the pore size as well by putting on additional layers. So for cell encapsulation, for example, you can put on a polyl lysine layer, and that can actually affect what materials come in and out. The pores are not massive in size as it is. So if depending on your application, for example, if you're going to use them for transplantation. Normally the pore size will be adequate to prevent things like antibodies attacking the cells, but it would allow nutrients in and out as well. And there's specific tests then you can do to test the pore size. So you can add them to dextrons or Prilling solutions, and you can measure the molecular weight cutoff. You can also then, I mean, the main thing is if you do really want to get tight control, it can be hard to get it because of their natural materials. But you can, as I said, you can actually coat them with different materials, such as polylysine or other materials which allow you to get tight control of the pore size.
Mm-hmm. Okay, then the last question. Accordingly, what is the weakness, according you or in general, of encapsulation technology?
Oh, the weakness. Okay. I mean, certainly one of the weaknesses of the previous was there's not a lot of companies that are doing large scale. But I mean, that's changing by the hour, or by the hour. Sorry, by the year. Another weakness would be that there's not a massive amount of polymers. There's probably there's probably about 15 or 20 that are used. A lot of them are natural, but again, that's increasing as well. And a lot of the problem with industry is there's not a huge amount of knowledge out there, available knowledge, unlike spray drying or fluidized bed. So a lot of companies love technology. They see it as a great technology, but there's a lack of knowledge to take it from the lab to industrial scale. And I briefly mentioned that a lot of companies who do get this knowledge tend to hold it in-house because it gives them such a competitive advantage in the market that they don't like to freely release it. So that's the whole thing of bringing it from the lab to industrial scale would be its problems.
Okay, then thank you very much, Michael, for answering these questions. I would like to thank you as well, Alan McDonald, for his presentation, and you, Michael. Excellent, good job. I would like to thank you, Michael Kerwick, as well, to be part of the webinar and introduce you guys. And I'd like to thank you to the audience for answering my poll questions. In this regards, we are at the end. No further questions were arising. And therefore, we will stop here the record and the presentation. In any case, you'd like to reach us, you have the opportunity to find us via the homepage. Thank you very much. Have a nice evening, afternoon, and morning. Bye-bye.
Thanks, Willie.
Thank you. Bye-bye.
Right. Thanks, Alan. Thanks, Michael.